The CLL pilot was essentially initiated in parallel to the two cancer pilots run by Genomics England, and it was very much to test the sample pathway and the robustness of the sample pathway for liquid cancers. We have been recruiting over 300 patients now. All patients are coming from clinical trials, national CLL clinical trials, and the samples are being collected as part of the, to the Liverpool Biobank, and then from then being forwarded to for sequencing. We haven't got any results yet; they are eagerly awaited. But we are hoping that by joining up the clinical trial results and the very deep clinical data and phenotypic data from the clinical trials with the whole genome sequencing data, that this will allow us really to identify new predictors of response to treatment. It's always a challenge to do that, but it's much easier to do if patients are homogeneously treated as part of a clinical trial, and if you have got the deep clinical data and also the response data that was collected as part of a clinical trial, it makes the interpretation of the whole genome data so much easier. So that's why in hematology we decided to go down this route of recruiting patients from clinical trials into the Genomics England program rather than taking all comers out of the NHS. You need a good 1,000 of patients before you can make any significant, can't draw any significant conclusions from the whole genome data, and that's partly because of the clinical heterogeneity of CLL. It's partly because of the biological heterogeneity. So before we can draw any definite conclusions, I think it'll be a few years from now. What we are hoping to see is what I've mentioned before, which is, we will be able to actually understand the significance of non-coding mutations. And out of, we know that, taking together, maybe at the moment, using TP53, SUM-HD1, RPS15, we can detect or predict 50% of patients who relapse within 2 years from chemoimmunotherapy. There are 50% that we can't predict. And we've done the exome, we looked at the whole exome, we've done lots and lots of other experiments. Now it's up to, I think, the regulatory regions in the genomes and maybe mutation signatures and subclonal heterogeneity that will define why patients don't respond to treatment. And so that's what I am hoping we will be able to see. We will also see changes in the germline that might be important for drug metabolism. So we know from, as treating clinicians, we all have patients who will be extremely sensitive to chemotherapy, and then we have patients who will get lots and lots of side effects from chemotherapy. And that must be because of different ways of metabolizing the treatments. And so again, we hope to be able to see, by analyzing the germline whole genome, why some patients respond better or not so well to treatments, and why they get more or less side effects.